with light reaction, and now we are going to study dark reaction, and we'll complete photosynthesis. As we have studied, during light reaction that is taking place in the thylakoid membrane, and we produce ATP and NADPH. As these thylakoids are present in chloroplast, these thylakoids are actually producing ATP and NADPH by the process of light reaction, and these ATP and NADPH now present in a stroma of a chloroplast. And within this stroma of chloroplast, another processes can take place. That process is said to be dark reaction. And we do also call this process as a light independent reaction as well, because in this process light is not required. And to respect the name of a scientist, Kelvin cycle, also we term it as a Kelvin cycle as well. What happens during dark reaction? Dark reaction is actually divided into three phases. There are three phases of the dark reaction, and out of these three phases we say the first phase is called carboxylation phase, and then the phase reduction phase, and the final phase that is called regeneration phase, where we are generating RUBP back. What actually happens during dark reaction? By consuming ATP and NADPH, we are going to fix carbon dioxide from atmosphere, and we are going to incorporate it in a triose form, so that this triose is stored in a stroma that can later help synthesize glucose, and this way plants can produce their food, sucrose or etc. How this actually happens? What is a process? How this carbon dioxide is incorporated into an organic simplest triose in form of glycerol aldehyde 3-phosphate. This is what the study. This is what the process that we have to study. Starting from the first phase is called carboxylation, and carboxylation is a phase where fixation of carbon dioxide will take place. In a stroma, there is a precursor molecule present that is called ribulose 1,5 biphosphate. Ribulose 1,5 biphosphate is already present in a stroma, and it has the ability to react with the carbon dioxide with the help of an enzyme called ribisco enzyme. And we will study later, ribisco actually is an enzyme that can work as a carboxylase, and it can work as an oxygenase as well. But during dark reaction, we study carbon dioxide reacts with the RUBP with the help of an enzyme called ribisco carboxylase, and this carbon dioxide is fixed in RUBP. And when carbon dioxide is fixed, the first step of a dark reaction actually takes place, whereas a fixation of carbon dioxide is taking place, and when this carbon dioxide is fixed in RUBP, they synthesize hexose. For example, if we are taking one RUBP, it is 5 carbon, and if we fix one carbon dioxide from atmosphere, we will synthesize one hexose. But as we are taking 3 molecules of carbon dioxide, we are reacting them with 3 molecules of RUBP, we would fix 3 molecules of a hexose. It is not just because we have to remove at least 3 molecules of a carbon out, incorporated in a simplest organic form, and the carbohydrates, as we have studied, they can exist in a simplest form, which is triose form. Therefore, we are taking 3 carbon dioxide, so that we can at least release a simplest form of triose in a stroma, which can later combine with another triose to synthesize glucose. Therefore, when we take 3 molecules of carbon dioxide that react with 3 molecules of IUBP, and they synthesize 3 molecules of hexose. And this is the first phase where carbon dioxide is fixed. Carbon is fixed, we say it is said to be carboxylation. Glucose is an unstable product. It immediately breaks into triose. And when it breaks into triose, finally we are having total 6 molecules of a. As we have 3 molecules of hexose, if they will break into triose, each hexose will break into 2 triose, and finally we are going to have 6 molecules of triose, each having 3 carbon, and to each molecule we name is 3-phosphoglycerate. It is said to be 3-phosphoglycerate. As these are glycerate and we'll have to convert them into aldehyde, therefore there is a requirement of another process which is going to take place, that is said to be a reduction, where we are going to reduce this glycerate into an aldehyde, where we are going to reduce ketose into aldose sugar so that we can finally synthesize glucose. And to this process, we simply say 6 molecules of a 3-phosphoglycerate synthesized during carboxylation phase will undergo a process of reduction. So this first process is to be understood and it's very important. 3 molecules of carbon dioxide fixed with 3 molecules of RUBP, they synthesize 3 molecules of hexose that immediately break into a 6 molecules of triose, and these are termed as 3-phosphoglycerate. As we have studied, during dark reaction, when carbon fixes, it first produces unstable molecule, and later when it breaks, it produces stable molecule. And if the stable product of a dark reaction contains 3 carbon dioxide, we term these plants as a C3 plants. We term them as a C3 plants. And there are plants which during this carbon fixation produce their stable product containing 4 carbons. We consider them as a C4 plants. And I'm going to produce a separate lecture for C3, C4 and CAM plants. I'm going to explain them in a detail later. But if hexose breaks into triose, we say these plants are said to be C3 plants. And this triose, 3-phosphoglycerate, undergoes a phase that is called reduction phase. And by this phase, what will actually happen? When we say this is to be reduced, something is to be oxidized. And on first step, we are going to oxidize 6 molecules of ATP, and they are converted into 6 molecules of ATP that remove 6 phosphate, and each phosphate combines to 3-phosphoglycerate. And we have finally 6 molecules of 1,3-biphosphoglycerate. Because the phosphate that's coming now comes to attach to a carbon number 1 and it synthesizes 1,3-biphosphoglycerate. And on the further stage, on the next stage, we are again going to reduce 1,3-biphosphoglycerate onward into glycerol aldehyde 3-phosphate. How? 6 molecules of NADH are oxidized into 6 molecules of NADP, and they are producing hydrogen, and they are reducing to 1,3-biphosphoglycerate into a glycerol aldehyde 3-phosphate. As we have phosphate on carbon number 1 also, so not only at this stage reduction of 1,3-biphosphoglycerate takes place, but the removal of a phosphate is also taking place. So what is going to happen on this stage actually, from 1,3-biphosphoglycerate, we will reduce 1,3-biphosphoglycerate with the oxidation of 6 molecules of NADPH. NADPH are oxidized and they are reducing 1,3-biphosphoglycerate. This is not only reduced, 6 phosphate molecules are removed as well. And resultantly, 1,3-biphosphoglycerate is converted into 6 molecules of glycerol aldehyde 3-phosphate. And these 6 molecules of glycerol aldehyde are now termed as a G3P. We term them as a G3P. This actually is an abbreviation which in a synth textbook is used for the first product also. This is what it is in a confusing, which is to be cleared. If you are considering this molecule as a G3P, but it actually is in a glycerate form, and finally again if you are producing G3P, which actually is an aldehyde form, therefore it confuses to the students. Therefore, we say this is said to be 3-phosphoglycerate molecule, whereas aldehyde form is referred as a G3P. Therefore, good to write G3P for a glycerol aldehyde 3-phosphate. And 6 molecules of glycerol aldehyde 3-phosphate are produced, and out of 6 molecules, 1 molecule of glycerol aldehyde 3-phosphate comes out of this cycle. Why is this molecule coming out? Just understand that these actually 3 molecules of carbon dioxide that we have taken, that we have fixed during a carboxylation phase, as by the series of process, we have converted them into a organic form, and to that organic molecule in form of triose, now we are releasing in a stroma, where they can be incorporated to help the synthesis of glucose. Therefore, as if we have converted carbon dioxide into an organic form by a process of dog reaction. And this 1 Kelvin cycle is producing 1 molecule of a glycerolaldehyde 3-phosphate. When 1 comes out, the remaining 5 molecules of glycerolaldehyde 3-phosphate, they are present again in a stroma, and they will have to regenerate the 3 molecules of a RUPP. Now try to understand, each molecule of a glycerolaldehyde has a 3 carbon, and there are total 5 carbon. When we multiply with the 5, we will again have a total 15 carbon. And similarly, we are going to produce 3 molecules of a RUPP, and 3 molecules of RUPP, and each RUPP has a 5 number of carbon. Again, we need a 15 carbon to regenerate 3 molecules of a RUPP. That means we have actual same number of molecules, same number of carbon dioxide. We simply have to rearrange the triose into pentose forms. And this rearrangement takes place by a process that is called regeneration. During regeneration, we are actually rearranging the triose form to a pentose form. And as if we are converting a glycerolaldehyde 3-phosphate to ribulose 1,5-biophosphate. And by this process, we say it is taking place at a phase that is called regeneration phase. And during this regeneration phase, we are going to consume 3 molecules of ATP into 3 molecules of ADP, and we are adding 3 phosphate. And each phosphate attaches to each molecule of RUBP and converts it into a ribulose 1,5-biophosphate. And by this way, glycerolaldehyde 3-phosphate, the 5 molecules of glycerolaldehyde 3-phosphate are converted into 3 molecules of RUBP, and to this cycle, to this phase, we said this is to be a regeneration phase. And this way, by these 3 phases, carboxylation and by reduction and by regeneration, we can fix carbon dioxide into a triose form, and to this cycle we say it's called Kelvin cycle, or it's called dog reaction. For just 1 glycerolaldehyde 3-phosphate, see, we are consuming 6 molecules this one and 3 molecules this one. We are consuming total 9 molecules of ATP, and we are consuming 6 molecules of NADPH. Therefore, we say 9 molecules of ATP and 6 molecules of NADPH are used just to produce a single molecule of a triose, that's said to be glycerolaldehyde 3-phosphate. And in order to produce 1 molecule of glucose, we will have to double this total amount of energy, and we'll have to repeat the cycle. Then we'll be able to produce 1 molecule of a glucose. This is said to be dark reaction, and this is the end of the process of photosynthesis. Now I'll be dealing with the C3 plants and C4 plants, and also with alternative methods of carbon fixation. Thank you very much. Subscribe my channel, share it with your friends also, comment, like, and keep learning with me. Thank you again.